All right. Now, before we go on, let's take a look at a major class of proteins that are widely used as intermediaries in many signaling pathways. Now, they're not used in all by any stretch of the imagination, and many of these proteins are also going to be involved in other things, as members of the family, and that's what we call the GProteins. GProteins are widely used in signaling pathways, but they have many other functions. Certain GProteins, for instance, are going to be used as some of the translation factors. We already saw one type of GProtein, RAN, that acts as a shuttle bus carrying things in or out of the nucleus. Still, other GProteins affect the microfilament cytoskel, stimulating it to grow and old microfilaments to get longer and longer. So we see all kinds of different uses for GProteins. But one of the major things that GProteins are involved in is numerous different signaling pathways. Now, GProteins are a diverse, what we call superfamily of proteins. Now, superfamily of proteins means a whole huge slew of proteins that all seem to have some kind of distant common ancestor. They may be fairly different from each other, but they do have certain features suggesting that they evolved from a common ancestor way back probably at the beginnings of multicellular life on this planet, if not even before that. Okay, now, so think of it. If any of you folks have like an annual or periodic family reunion, we get people coming up from all over different parts of the South and different parts of the country, and they all gather together and get to see your 14th cousin for, once a year. You ever have those things? Okay, all right. That is a superfamily. You may be very different from your third cousin who comes from Cleveland or something like that. Did I get that right? Okay. You may look different, different personalities, different lifestyles, whatever. But if you go back far enough in your ancestry, there will be a common ancestor. Somebody, two folks had some kids, and their kids had some more kids, and goes down, and then all of a sudden you get this family, this extended family of hundreds of different people. Okay, that's what a superfamily in proteins are. They have a distant common ancestor. They may be fairly divergent, and there's a lot of them. Many of them specialize for particular different roles. Okay, so GProteins fall into a big superfamily of proteins. Now, if you were to look at do genetic testing on that big extended family in the family reunion, you may well find certain genetic markers that are common to your family but rare or absent in others. In fact, there's a controversial thing about taking a DNA sample from somebody who is not necessarily a suspect in the crime and then thinking they have the DNA sample at the crime scene, trying to say, oh, well, this person is related, this culprit is related, and then they try to pressure everybody in the family to get tested to see if they can get the culprit. That's happening in some states, and that's viewed as extremely controversial and stuff. It's going to end up in court one of these days. But at any rate, you would find, if the cops can do it, so can protein evolution. You would find that there are certain features in common in these superfamilies of proteins. Now think with the GProtein superfamily. They all have certain important characteristics. All of them bind a guanine nucleotide. In other words, either GTP, guanosine triphosphate, or GDP, guanosine diphosphate. Just going to shift the position a little bit on this. Okay, it's probably a little better. Okay, so all of them bind a guanine nucleotide. All of them have two different structural states, depending on whether GTP is bound or GDP is bound. So we'll put different So if you got GTP bound to the G protein, it has one structure. We could represent it, for instance, as a square. If GDP is bound, it has a different structure. We'll represent that as a circle in this case. So just by looking at the structure of that protein, you can say whether it has GTP or GDP bound to it. So that's another common feature of all G proteins, whatever the function is. Now, third feature. All G proteins are GTPases. In other words, they hydrolyze GTP into GDP and phosphate, and they release the phosphate. Now, they're not very good at that. They're not very good at GTP hydrolysis. They're actually quite slow at it, but they will do it eventually. So that means if you're a G protein, you have GTP bound, what's going to happen to you? Eventually you will hydrolyze it, and then you'll go into the GDP state. So all the G proteins are weak GTPases. Okay. All right, now another thing. All G proteins are able to exchange GDP for GTP. Okay, now, this is an exchange. A G protein cannot make GTP out of GDP. They simply can't do it. Does anybody know why that would be the case? If you can take GTP and hydrolyze the phosphate off and turn it into GDP, why couldn't you go in reverse? It requires a condensation reaction. Condensation reactions are endergonic. They take energy. In fact, to do that, to make GTP, that takes a lot of energy. They simply can't pull that off. Now, I give you another analogy about this exchange process. I put this in because people sometimes do make mistakes on it. Give you another analogy about exchange process. Okay, well, you know, I didn't do it this time. My wife's birthday's on Valentine's Day. Now, let's suppose I want to go and get her, as a sort of enlightened self-interest, to get her a nice, snuggly nightgown, right? Okay? So, all right, well, I go to the store, and of course, I'm not very good at shopping for women's clothing because women's clothing is so bizarre. But let's suppose I want to get a nice, fuzzy, snuggly nightgown. Good for winter, good for her, good for me, whatever. Okay. Now, go down to the store, right? Now, I don't quite know what her size might be. I have an idea, but I'm not all that good at it. So the question is, what size should I get? Should I get a medium? Should I get a large? Whatever the case is. Well, being a fairly prudent person, I may want to err on the side of caution, get the smaller of the two potential sizes, so I don't sleep on the couch for a month afterwards. Okay, so, all right, now let's suppose I do this. And I get the smaller of the two sizes. And I get it to her, and she opens it up, and oh, this looks so cute, you know. And she puts it on, and can't quite fit it. Okay, well, I was cautious, but she can't wear it. It's too small or too tight. So what's she going to do? She takes it back to the store and exchanges it for hopefully the same one and a somewhat larger size. What does not happen is she takes it to the store and the clerk looks at it, oh, this is too small, pulls out a pair of scissors and cuts a slit in it, and then gets a piece of fabric and sews it on to turn the smaller one into the larger one, because that takes too much time and energy to do it. So instead you do an exchange. That's what we're seeing with the G proteins. They exchange GDP for GTP. They cannot turn GDP into GTP. Now these are common features all G proteins have. All of them have these features in common. Okay, now, let's take a look at a couple other aspects of G proteins. we can lump the G proteins into two broad categories. One of them is what we call the monomeric. Now, the term monomeric refers to single subunit. In other words, it's a single amino acid chain. They often call the monomeric ones the RAS. RAS is a major one involved in a critical signaling pathway superfamily, because they're all distant relatives of this one G protein called RAS. RAN, the one that shuttles things back and forth from the nucleus to cytoplasm, is a member of this. So monomeric G proteins have a single subunit. But we also have the trimeric, or often called the hetero. Hetero means different. So I'll even put the term hetero. You'll see both. Okay. What that means, trimeric means 3 subunits. And hetero means different, so there's 3 different proteins assembling together to make these types of G proteins. Okay, now the monomeric G proteins are involved, some are involved in signaling, but many are involved in other things. We mentioned things like RAN, nuclear cytoplasmic transport. There are these G proteins that are going to be involved in regulating the microfilament or actin cytoskeleton, in terms of generating cell extension, cell movement, stuff like that. And there are still other things. So the monomeric ones, RAS is important in a very critical signaling pathway, but there are other members of the monomeric G proteins that have nothing to do with signaling. They play other roles in cells. Now, the trimeric ones with 3 different subunits, all of them are involved in signaling pathways. They're exclusive, as far as we know at least, to signaling only. They're specialized for that. Now let's take a little look at these heterotrimeric ones, these trimeric G proteins. We're going to have 3 different subunits. I often draw it kind of like the old style P symbol. And crudely, tertiary structures, crudely, or quaternary structures, crudely like it. We have 3 different subunits. One of them is called alpha. And that's the one that binds the guanine nucleotide. We'll put G. Now, the alpha subunit is actually structurally quite similar to the monomeric G proteins, and clearly it probably evolved from one and then had the ability to bind to these other 2 subunits. And there's another subunit, the beta subunit and the gamma subunit. Okay, the beta and gamma subunits don't bind guanine nucleotides at all. In fact, a lot of times they don't do too much except keep the alpha subunit attached and in an inactive state. As we'll see later on, when the alpha subunit binds GTP, the structural change causes it to fall off the other 2 subunits. And many times the other 2 subunits sit there staring at you. They do have some other functions too. But for the most part, the beta and gamma subunits, the dirty work is done mostly by the alpha subunit. Yes?
Does that say GTP or GDP?
Oh, I put GDP down here. And the reason why is that when it's in the GTP state, the structure change causes it to fall off the beta and gamma subunits. But that comes later. So I just want to keep some consistency here. Okay, so we have these kinds. These are the trimeric ones. These are exclusively used for signaling. There's a reason why. Each one of these is going to recognize one or more receptors, and when it's activated, it's going to ultimately do some kind of regulation, whatever it is. Okay, well, cool thing about these, and this is one reason perhaps why they're so widely used for signaling, is there are about 10 different genes for each one of these subunits. There's several different versions of each subunit. So there's, I think there might be 14 alphas, and then there's 6 or 9 betas or gammas or something like that, but currently about 10 different versions of each subunit. And a functioning trimeric G protein, to make that, all you need to do is take one alpha subunit, one beta subunit, and one gamma subunit. You can mix and match any alpha and any beta and any gamma, you put them together, and they make a trimeric G protein. What that means, if you have about 10 genes for each subunit, the number of possible trimeric G proteins you can make is roughly 10 times 10 times 10. In other words, roughly about 1,000 different trimeric G proteins. Each one of them will recognize certain types of receptors and give certain types of cellular responses.
So you said you can do 1,000?
Yeah, so we're up 10 cubed, 10 to the third power, yeah. That's approximate. Okay, so what it means is with a mere 30 genes, you can come up with 1,000 or so different G proteins. And that means a lot of different kinds of receptors can use these trimeric G proteins as intermediaries. So this can support many different signaling pathways and many different receptors. So that's one reason why these things are so widely used in many different signaling pathways. Now, not all of them, but they are used in quite a few. And we'll see some examples of that later on. Okay, so that's the trimeric or heterotrimeric G proteins. Now let's take a little look at our sort of cycle of a typical G protein. We saw a little bit of this before when we looked at RAN, but let's take a look at this. First, we're going to look at the activation and shutoff cycle of the monomeric G proteins, and then we'll look at the trimeric ones, which have, because the other subunits have a slight variation. Okay, now what I'm going to represent is, for the monomeric G proteins, a circle is going to represent the G protein with GDP bound to it. A square will represent the GTP bound state. That tells you we've got different structures here, different tertiary structures here. So we start out using the monomeric. Okay, we're going to start out here with the G protein in the circle because it has GDP bound to it. In order to activate this G protein, at least switch it to the other state, what we have to do is this exchange. GDP for GTP exchange. So, in this case, the G protein expels the GDP, and in comes a GTP. And having done that, now it undergoes the structural change. Okay, now this exchange process is normally quite slow. It doesn't happen very often. The G protein binds GDP in this binding site fairly tightly. It's not too likely to shake off on its own. Now, on the other hand, GTP, if it can get in, will actually bind to that site even more tightly than GDP does. So once in a while, the GDP will shake itself loose, pop off the G protein. If there's a GTP around, it'll go in that site and bind it tightly enough that GDP wouldn't be able to displace it. Problem is, this takes a long time. So what we can do is stimulate this process with these proteins called guanine nucleotide exchange factors, or GEFs. I'm going to put one right over here to stimulate this process. And we ran into this with RAM once again. Okay, and what these basically do is they speed this exchange process up 1,000-fold. What would ordinarily take on the average maybe once every 10 minutes or so can happen in less than a second. Now what these proteins do is they bind to the GDP form of these monomeric G proteins. They warp the binding site in such a way that the GDP can fall off fairly easily. Now when a GTP comes in, it grabs on site tightly enough, causes the structural change in the G protein, and that structural change causes the exchange factor to pop off the G protein. So that's how these things work, by binding and basically warping the binding site that holds the guanine nucleotide. So now the GDP falls off real easily. Okay, so now we've got this. Generally, this is the active state. I'll put a little star here to show that. And what it does, what G proteins do when they're active, is they bind to and regulate numerous target proteins. There can be all kinds of different ones. Okay, now, they can do this repeatedly. So here's where we're going to get some amplification. The active G proteins binding and regulating numerous target proteins. But all good things must come to an end. G proteins are GTP hydrolyzing enzymes. They're GTPases. So eventually they're going to start doing their thing, GTP hydrolysis. So I'm going to put kind of like an intermediary state here, where we have the GDP and the phosphate, but we haven't let it go yet. And then we kick the phosphate off. And we switch back to the original GDP-bound state. Now, we said that G proteins are GTPases, GTP hydrolyzing enzymes. The problem with them is they're really crappy at it. They're very weak GTPases. On the average, with one of these G proteins, it'll take a full 10 minutes before it finally gets off its butt and actually hydrolyzes the thing. So they're very weak GTPases. But we can speed that process up with a second set of regulatory proteins, the ones called the GTPase activating proteins, or GAPs for short. Now what these guys do is they bind to the appropriate G protein and they warp the active site that does the GTP hydrolysis. But they warp it in such a way that the active site is now orders of magnitude more efficient than it used to be. So when one of these guys bind, it causes the GTP hydrolysis in less than 1 second. By the way, these are really interesting examples of how one protein can regulate another, and basically what it does. In this case, these GTPase activating proteins warp the active site of the G protein when they bind and make the active site far better than it used to be. Okay, so either way, so we have these regulatory proteins that stimulate the exchange and activation, and stimulate a different set that stimulate the GTP hydrolysis and inactivation. And that's the cycle we see for the monomeric G proteins. Okay. Now let's take a look at our trimeric G proteins. The trimeric G proteins have a fairly similar mode of action. The big difference is now we have those two other subunits, the beta and gamma subunits, that we didn't have with the monomeric ones. Myra, question?
Oh, no.
Oh, okay. All right. Okay, so let's look at the trimeric ones. Fairly similar kind of thing. Don't forget the alpha subunit is very similar structurally to the monomeric G proteins. But let's take a look at these guys, because of course they're involved in signaling. All right, now what we're going to do, it's the same kind of cycle, but we're going to do a few modifications here. Make the proteins a little bit different. All right. Okay, we're going to start out now with a trimeric G protein in the inactive GDP bound state. Once again, I'll represent that as sort of like a P symbol here. Alpha here, beta, gamma. And here's GDP. Okay. Now, it too can do a GDP for GTP exchange. And there are their own sets of guanine nucleotide exchange factors, GEFs, that can speed that up. As a matter of fact, what we're going to see is these guys. It turns out that occupied receptors are the GEFs. So the G protein binds this receptor directly, and it stimulates that exchange process. Without the receptor, the exchange process happens very, very slowly. So we'll have this sped up over here. Now what we're going to do is we're going to do that exchange process here. Out goes the GDP, in comes the GTP. Okay, now when that happens, the alpha subunit undergoes a dramatic tertiary structure change. And so they actually have that, by the way, in the literature. And structural change is quite impressive when you see it. It almost looks like two different proteins. So now what's going to happen is this. The alpha subunit undergoes a structural change that causes it to fall off the beta and gamma subunit. And the beta and gamma subunit now basically separate off from the alpha subunit. The alpha subunit goes and binds to various target proteins. The beta and gamma subunits, many times they don't do anything. They just sit there staring at you. In some cases, though, the beta and gamma subunits, it depends on the G protein, the type of G protein. But in some cases, the beta and gamma subunits, separate from the alpha subunit, can bind to and regulate proteins in their own right. That's less common, but it can and does happen with some certain G proteins. Okay, now, the active G protein, just like the monomeric ones, the active alpha subunit is the thing that does most of the regulation of target proteins. Once again, it too is a GTPase. It will hydrolyze the GTP, although very slowly. So put it partway in here. Okay. That process can be sped up by their own GTPase activating proteins. That speeds the process up. Once we have done that hydrolysis here, we got our alpha subunit back in the GDP state. And now the beta and gamma subunits come back together and rebind to the alpha subunit, kind of dock with it. And we're back to where we started from. So the major difference here is that when the alpha subunit binds, has GTP in it, it can no longer bind to the beta and gamma subunit. When it hydrolyzes GTP into GDP, now it can rebind to those other two subunits. So that's a major difference here. Now, G proteins play all kinds of different roles. As a matter of fact, a couple nasty bacterial toxins work by inhibiting specific alpha subunits. Now, and are even used as research tools. That's what you want. Okay.
Yes, sir.
All right. Case in point, cholera. Cholera was a pretty big killer. It still is in places with poor sanitation, like refugee camps and stuff like that. The cholera was the nemesis of Europe for a long time. Because cholera is spread through feces. And if your cooking and drinking water is contaminated with human waste, that's a great way for it to spread. Now the cholera, it turns out that cholera can kill you in a matter of hours untreated. It turns out it's not so much the bacteria itself that kills you. What it does is it secretes a protein toxin called cholera toxin. What cholera toxin is an inhibitor of a specific G protein that's found in the cells in the large intestine. It blocks the alpha subunit, and when that G protein is ordinarily active, what it does is open up and stimulate transport of ions across membranes. The ion transport leads the cells in your large intestine to pick up water, so that way what goes through you, you absorb most of the water, and then a semi-solid mass comes out the other end. But inhibited by cholera toxin, the large intestine can no longer absorb water, so everything comes out. The cause of death in cholera is dehydration. You lose too many salts and electrolytes, and you lose too much water. And you can dehydrate fatally in less than a day. Treatment for it isn't antibiotics. Treatment for cholera is rehydration. They give you either drinking or by IV, usually by IV, saline, nutrients, and lots of water. If they do that, you will pull through. You will feel miserable for many days, but you will pull through and you'll come out none the worse for wear. If you don't do that, you'll be lucky to last 2 days. So cholera toxin works by inhibiting one of the alpha subunits specifically. It just so happens. Now, in evolutionary terms, that's great for the bacteria. You are a fecally spread bacteria. So it helps your spreading your genes around if your host happens to hit the bathroom more often than usual. So if you put this toxin that allows this massive diarrhea, people can often end up losing it 50 times a day. Well, that goes into the watersheds, into the environment, and it makes your spread a lot easier. That was a real big problem in the European cities because they didn't have much in the way of sanitation. So, you know, people, of course, they didn't have running water or anything like that. So what happens, people would dump in their chamber pots and pee and dump in their chamber pots, and then they would dump the contents out of the window and into the street. In fact, that's where the old tradition, the old sort of etiquette tradition of the guy walking on the outside and the sidewalk came from. It wasn't so much cars or anything like that. It was that many buildings in Europe, the second story would overhang several feet from the first story. You can go to European cities and still see that. So what happened? You're walking down the street, the guy on the outside walking down the street is more likely to get hit by the contents of somebody's chamber pot, because don't expect these people to bother to look out the window before they dump their contents. But the problem is, that stuff would run down the streets into the local watersheds, the same water that you use for drinking and cooking and stuff like that. So cholera outbreaks in those densely populated European cities were devastating. They killed thousands and thousands of people. They'd sweep through the city in a short period of time. Nowadays, with better sanitation, now interestingly enough, you didn't have so much with the Romans, because the Romans actually had for their days, a fairly sophisticated water and sanitation system, so it kept cholera outbreaks down quite a bit. Europe was not so much the case. A few things the Roman Empire did that was good, and that was one of them. Also, when there were cholera outbreaks and things like that, Jewish people who tended to be just following the kosher laws, more sanitary, often were less risk than the average population, which often meant in the days of Europe in those days, people would accuse the Jews of poisoning the wells and stuff, and then they'd go out and murder them. And they did plenty of that in Europe in those days too. So yeah, there was a lot of that kind of stuff, but cholera was a very serious problem here. Nowadays you hear of outbreaks, and especially places like refugee camps. They had a real problem with that in Haiti after that massive earthquake and stuff. Huge amounts of cholera spreading all over the place in those refugee camps. So that's the kind of thing, but it shows you the importance of just one alpha subunit, and inhibit that causes some real serious problems. Tetanus also produces, the tetanus toxin inhibits a different alpha subunit, leading to the catastrophic muscle contractions that cause tetanus. So that's another kind of thing. So it's just a couple examples of G proteins, trimeric G proteins, and signaling and physiology. Okay, well that's what we got here. We've taken a look at their G protein. We're going to start looking at individual signaling pathways when we come back on Thursday, and see you folks in the lab fairly soon, and try to get things up on YouTube, and at least get the processing done. Okay, folks.